A very good evening to all of you. My name is Prithvi Singh and I'm working as a PhD scholar at the Center for Interdisciplinary Research in Basic Sciences, Jamia Millia Islamia in New Delhi, India. The topic for my poster presentation is Integrative Multiomics and Weighted Network Approach reveals the prognostic role of RPS7 in lung squamous cell carcinoma pathogenesis. An SCLC can be categorized into lung adenocarcinoma, LUAD, and lung squamous cell carcinoma, which is LUSC. The 85% of cases are being subjected to LUAD. The risk factors include commonly as smoking, radon exposure, family history, and the 5-year survival rate of an SCLC patients has been quite poor and declining despite radiotherapy, chemotherapy, and various targeted therapies. LUD occurs mostly in smokers, women population low carcinoma exposure, whereas LUC occurs mostly in men, smokers, and have a larger tumor size compared to LUD. LUC is known for comparatively less typical oncodrivers, target therapy resistance, marked genomic complexity, and a higher mutational rate. The methodology which we followed primarily involved the data extraction pertaining to LUAD and LUC patients from UCC synaptoses, followed by their cleaning, pre-processing, and differential express genes identification as compared to normals. The weighted gene correlation network analysis and upgenes identification, protein action, and overall survival analysis was done in the second section, followed by validation using external databases such as cBioportal for marking the mutational complexities, ULCAN, and Human Protein Atlas. The differential expression analysis, as shown, the results showed that more female populations were there in LUAD as compared to LUSC, which comprises more of male populations, and this goes well in trend with the theoretical concept. The DEX, the ratio of DEX was 60% LUSC and 40% were for LUAD. We obtained a total of 1538 differential express genes in LUAD and around 2364 differential express genes in LUSC patients. The weighted gene expression correlation network analysis, both LUAD and LUSC, were done by primarily removing the noisy genes prior doing the WGCNA, for which we obtained a total of 944 LUAD associated differential express genes and 588 LUC associated differential express genes. These were subjected to the weighted gene co-expression network analysis, and in the LUAD we found four different highly correlated modules: blue, brown, yellow, and turquoise. The blue module had 203 genes, brown had 194, yellow had 95, and turquoise had 452. Whereas in LUSC we obtained a total of 5 modules: blue, brown, yellow, turquoise, and green. Blue had 56 genes, brown had 51, yellow had 45, turquoise had 402, green had 34. Now, based on the module membership and intra-module connectivity, which are an integral part of network centrality measures in WGCNA, we found the highest correlation, which were significantly also higher in turquoise in case of LUAD and brown in case of blue in case of LUSC. Now, all the genes from the turquoise module for LUAD patients and all the genes for the blue module in LUSC patients were subjected to the protein-protein interaction network analysis. A total of 42 genes and connected, which were connected by 327 nodes, were shown for the LUAD cohort patients, and a total of 18 genes connected by 138 regions in case of LUSC were shown for the LUSC patients in case of the PPI network. Now, when we did the survival analysis for both these PPI network genes, we found that only two, which is RPS7 and RPS15, were significant in both the lower and higher cohort patients, as we can see in the overall survival analysis Kaplan-Meier graphs. The prognostic analysis showed that RPS7 had a nearly double mutational amplification frequency, which is 1.2% as compared to RPS15, which is 0.6%. Both were having a high amplification frequency, and the patient samples which we chose in cBioportal were the exactly same as we retained for the differential express genes analysis. Also, there was a high correlation of 0.74, which is significant between RPS7 and RPS15, shown by the scatter plot. And The RPS7 had a significant difference between normal and tumor patients, as shown by the box and whisker plots. The Human Protein Atlas showed that in the normal lung patients, the RPS7 had a comparatively higher macrophage content as compared to the lung squamous cell carcinoma, in which the macrophage content decreased, showing that RPS7 was downregulated. Both RPS7 and RPS15 expression status were downregulated. The concluding remarks showed that RPS7 had a higher mutational frequency than RPS15A, and RPS7 can be linked to the ribosomal stress induced P53 activation, and it also decreases the content of metabolic proteins such as GLUT4 and various other proteins which are required for exhaustively increasing the cell proliferation in cancer. RPS7 silencing or downregulation can lead to increased cellular proliferation via dysregulation of cell cycle checkpoint proteins and cyclin-dependent kinases such as CDK1, 2, 4, CCNB1, CCND1, and it is correlated with SMYD2 and PIM1 also for its downregulation. It is the first kind of study which we are showing in case of lung cancer, which has not been reported earlier, as uh, irrespective of its downregulated status. It has always been shown as an upregulated protein in many cancers, such as colorectal cancers, but this is the first kind of study which needs further wet lab validation to be proven. Thank you, and I close my presentation.